Today with quorum sensing, we're going to answer three questions. Can microbes communicate with each other? And if so, how? What are some of the languages that microbes use? How are they different? And who do they allow a microbe to communicate with? And what are some of the uses of quorum sensing? So a quorum essentially, at this basic level, is the minimum number of members of an assembly or society that must be present at any of its meetings to make the proceeding of that meeting valid. So essentially the size of the group that is needed to accomplish a given task. Quorum sensing is cell-to-cell communication that enables collectively modified behavior in response to changes in cell density and species composition of the surrounding microbial community. So essentially, this is the ways that microbes can tell how many cells are around, and if they have changes in the cells that are around them, both in density or composition, they can then respond. And so as we've been sort of talking about through this course, bacteria do not live in isolation. They don't live just as one cell. They don't live as a single species community. They live in highly diverse and highly abundant communities. And it doesn't really matter where you are, whether it's the extreme environments we're going to talk about next week, or if it's on the human body. Bacteria don't live alone. They live in high abundances. That being said, because of this, many of the actions of bacteria have to be coordinated, and the behaviors are typically depending on how dense the cells are around them. And essentially, they do this by talking with each other via a chemical communication. So we're thinking about microbes as a whole. There is evidence that archaea also perform this quorum sensing, this chemical communication. But we're actually going to focus on bacteria today simply because bacteria are much, much more heavily studied. And as you'll see, much of what we know about quorum sensing is actually in a medical setting. And as we've talked about, there are no known archaeal pathogens at this given moment. And more importantly, I think from an evolutionary perspective, this seems to be the precursor for multicellular organisms. Both us and even the more simple things like, say, a sponge or a jellyfish. We all know that multicellular organisms are simply just a collection or a mass of cells working together, and they have to coordinate things sometimes across large distances. So you can think about if you're trying to communicate with your brain to your feet, you have to send a signal, maybe 5 to 6 feet for this to work. And quorum sensing is very much the same way, but it just at that microscopic scale. These bacteria and, again, potentially archaea need to send a signal a few hundred microns to make things work. And so this is really a precursor to how we do things at the trillion cell level. And so there's a very basic mechanism for how quorum sensing works. It starts with the production, the release, as well as the detection of extracellular signaling molecules. These molecules are what we call autoinducers. These autoinducers build up in the environment as the bacterial population becomes more dense, i.e. Having many more individuals in a given area. And essentially, these quorums, these autoinducers essentially act as a proxy for the number as well as the density of cells, because if you have more cells or you have denser cells, you're going to have much, much more autoinducers in the environment. And then finally, once you achieve a certain concentration of autoinducers, the cells collectively alter their patterns of gene expression and thus their behavior. So there are three key parts to quorum sensing that we need to understand. So we're just going to outline the basics of them right now. First is an enzyme that makes the autoinducer. This is typically denoted with an I in its name. So we typically see things like LuxI or RPAI. We need the autoinducer itself, or the AI for short. These can be different molecules, but typically speaking, gram-positive bacteria do produce peptides, which are just short carbon-based molecules, and our gram-negatives typically produce N-acetylhomoserine lactones, or AHL for short. I will never call them N-acetylhomoserine lactones again, because that is a mouthful, so we'll refer to them as AHL going forward. One of the things that's nice about AHLs relative to peptides is that they do freely diffuse through the bacterial cell membrane and cell wall, so it allows them to get inside, outside of the cell with relative ease. And then finally, we need a receptor that ultimately binds to this autoinducer, and it is typically denoted with an R in its name, so LuxR or RPAR. As you can see, they match between these autoinducer producing enzymes as well as the autoinducer accepting enzymes. But ultimately, once an autoinducer is bound to its receptor, it initiates some sort of transcriptional change. And it also initiates the production of more autoinducer producing enzymes. Thus, it creates a positive feedback loop. And so we can just show a basic diagram here where we have a cell that is that would be in an environment that is very low density. So we're looking at HSLs, these homoserine lactones for our gram-negative bacteria. And you notice that there are very little concentration here on the left. And thus, there's not very much being produced by LuxI, which is the autoinducer producer, and LuxR, which is the autoinducer receptor, is not being bound. But what you'll notice on the second panel over here, where we have many, many more HSL signals, we're producing a lot via this LuxI protein. The LuxR protein, which again, that receptor is being bound, and then ultimately it's binding to the DNA, inducing transcriptional changes, and thus a change in the behavior of these microbes. So we're going to focus most of our talk today about gram-negatives simply because they are much better studied, but we'll talk briefly about gram-positives for a moment. And so a gram-positive mechanism is through the use of peptides. So in this figure over here, we have these green pentagrams. Remember, these are in substitution of those AHLs we see in gram-negatives. Essentially, it has a response regulator that gets phosphorylated by the transmembrane receptor. So we have this receptor that's embedded in the membrane over here, and under high enough autoinducer concentrations, it will get bound, and then it will phosphorylate this regulator on the inside of the cell. And then once this regulator in the inside of the cell is phosphorylated, it will then induce transcriptional changes. And so this is sort of the basic mechanism of what is called a two-component system. And it's particularly nice because without this two-component system, the peptide physically cannot enter the cell. It can be exported, but it physically cannot come back in unless this sort of outer membrane protein is bound. And this is called a two-component system because it has two components. So we have the receptor as well as the inside protein. Gram-negatives are pretty similar, and this is typically what we think as the sort of concrete basic example of how quorum sensing works in a cell. But ultimately, remember these autoinducers, these HSLs, they're going to be small organic molecules. It is typically an AHL, so that acetohomoserine lactone signals, but they can be many, many other types of autoinducers, and we'll talk about them very shortly. The idea being is at very high cell densities, it activates LuxR or any other receptor protein, and then it initiates transcriptional changes. So the process is very simple, very similar between gram-positive and gram-negatives, but remember in gram-positives it is that two-component system where you have to have a transmembrane protein, whereas in our gram-negatives, these HSL molecules will freely diffuse through the membrane in the cell wall to get inside. So there's no physical need to move them in and out. Our AHL autoinducers typically, well, not typically, they always pretty much follow the same sort of basic structure, same basic pattern. So they have this sort of core homoserine group, and then they have what we call an R group. And so you'll remember this from amino acids. All amino acids have this sort of the same core group as a whole. And the amino acids themselves differ by their R group. And that's exactly the same for our AHL inducers. You'll notice that this R group is what varies, and this R group can be sort of variable within a given species or be very different, but they're ultimately very differ across all sorts of different groups. And this is just sort of a small collection of different AHL or HSL autoinducers. And sort of the important thing to note here is that they're all very, very different, and thus they have very different protein targets they can bind to, thus making many of these AHL autoinducers species specific. And we're thinking about this, they're very specific, so we're looking at communication only within a species. It's essentially your own language. So you can think of this as like, each bacteria is from a different country and they speak a different language. So, this autoinducer is that speaks Spanish, and this one is French, and so on and so forth. And so it basically gives you your own species-specific language, and other microbes physically cannot pick up on your signal. That being said, there are many, many other types of autoinducers. The AHLs are the most common, and they're very, very specific. And there are variants spread across hundreds and thousands of taxa. We have atypical ones, which would be B through H on this, this graph here, or this figure here on the left. They're taxa specific. So for instance, D here, we have CAI1, which is specific to cholera. And then we have autoinducers 2, or the AI2, which is going to be in E over here. This is something that's very similar. It's found in greater than 500 taxa. And this is sort of a more broad, not really broad spectrum, but sort of broad acting type of autoinducer that allows from within and as well as between species communication. And so just to sort of highlight one clinically as well as environmentally very important organism is Pseudomonas. So we've worked with Pseudomonas in the lab a few times thus far. It is a gram-negative bacterium. It is important both clinically and as you will see next week is very important environmentally, in particular in soils. But Pseudomonas as a genus has multiple quorum sensing pathways. The vast majority of their autoinducers are AHLs, but they are not all like that. And what's sort of the interesting thing about Pseudomonas, it has multiple quorum sensing pathways, but oftentimes these pathways are interconnected with one another, which allows these organisms to coordinate multiple functions at the same time, whereas very many AHL autoinducers will only promote the function of one thing in particular. So when we're thinking about interspecific communication or communication within a given species, they can talk to any given species using their own unique language. But many structures, especially biofilms, as we'll talk about later in this lecture, are the coordinated efforts of multiple different species of bacteria. And so, for instance, when you're thinking about your dental plaque, so when you haven't sort of brushed your teeth in, say, maybe 12 hours, you get that sort of gross, filmy stuff on your teeth. That's a biofilm. And those dental plaque biofilms can actually reach densities of 400 different species at any given time. So it can be particularly dense. And so taxa can talk within a single species, but they can talk between species. And so when we're thinking about interspecies communication, we're thinking about something that's a common language. And so we'll use an example of Vibrio harveyi. Has multiple autoinducers, very much like Pseudomonas aeruginosa, but it has different receptors that will bind. And essentially, they work on different. Each of these AHLs will bind to their own unique their own unique protein receptors, and they will. And the sort of interesting thing is that even though they're different and they bind to different proteins, they actually activate the same pathway. And so what's sort of interesting about Vibrio harveyi is that under low cell densities, it activates these three pathways of quorum sensing will activate one set of genes, and then under very high cell densities will activate a completely different set of genes. And so this is a two-component system that we commonly see in bacteria, less so in archaea. But essentially, it's a sort of a very common system, as we saw with our gram-positive bacteria. It includes a transmembrane protein that senses something on the outside and then relays that information on the inside. And at very low densities, this LuxU is phosphorylated, and what this leads to is the production of APHA that leads to virulence in biofilm and shuts off bioluminescence. At high density, what ends up happening is the presence of AHL inducers in high densities dephosphorylates LuxU, which in turn then turns off virulence and biofilm formation and stimulates bioluminescence. So the question we can ask is, why do we have three signals for the same pathway? And the very short answer to this is that it allows them to talk to other taxa. So HAI1 is a typical AHL, but it's very specific to Vibrio harveyi, so it only really allows Vibrio harveyi to communicate with itself. However, EAC8CAI1, that is a mouthful and a half, is a very different type. So this is our CAIs. Here is our sort of our other type of autoinducer here. And what we're seeing is that this receptor is found in Vibrio harveyi as well as Vibrio cholera. So this is what we call intergenera communication, which allows this Vibrio harveyi to communicate with other type of Vibrios. But as you notice, we have an additional autoinducer here, AI-2. And AI-2 is over here. And the sort of interesting thing, AI-2, is that it's found in hundreds of taxa. It's found across multiple phyla, both gram-positive and gram-negative, suggesting that AI-2 is a very, very old system in an evolutionary sense. It allows for interspecific communication, so communication between even distant related taxa. And it also potentially might allow interkingdom communication, so communication between these organisms and potentially something like a fungus or a protist. But ultimately, quorum sensing is ultimately, we need to think about it like a circuit. And so many pathways under quorum sensing are parallel. And they might be essentially induced by a different autoinducer here. But they ultimately promote the sort of the same response. Other pathways are in what we call a series, and we would be more sort of thinking about as a circuit here. So the idea being was we have two different AHL and autoinducers here, and they both activate different signaling properties, and they essentially control two different processes on the same set of genes. So maybe this one activates something for, say, motility, and this one activates something for, I don't know, the production of flagella. And so what this does is allows them to potentially control things both temporally as well as spatially within a cell by having this on a circuit where you can do one thing with one autoinducer and do something else with another autoinducer. So that's sort of the basics. So bacteria communicate with each other, and these pathways can potentially be complex and involve both within and between species communication, and there's something that is particularly very old. And the question we should then ask is, why do we care? And the important thing to remember here is this is important clinically because it allows microbes to do things that they wouldn't be able to do alone. And so one of the things that quorum sensing allows bacteria to do is actually evade predators. And interestingly enough, many mechanisms only work when there are in high enough densities of cells to have an effect. So for instance, Chromobacterium violacin, this picture doesn't really do its purple pigments any justice. They're very beautiful in person, but Chromobacterium actually produces a toxin called violacin, and violacin is very toxic to both bacteria and eukaryotes alike. However, this is actually only turned on when cells are in very high density, and what this does is by keeping it off most of the time, it allows these cells to conserve resources as a whole, and by sort of delegating this in terms of having sort of a communication between many different cells allows others to, within sort of this community, to save resources while still getting the benefit of predator defense. The other way, another way that this process of quorum sensing is useful is in the production of biofilms, which we will talk about later in this lecture. But briefly, right now, biofilms are highly organized consortia of typically multiple bacteria, but they can't be a single species. And the formation of a mature biofilm, as we're talking about, does require the coordination of all the members of the community, typically releasing exopolysaccharides, which you can see in this picture over here, sort of this gooey stuff in between all these different cells, which allows these biofilms to be sticky. But ultimately, the communication between this sort of conglomeration of cells is typically done through quorum sensing pathways. In addition, quorum sensing is important for virulence. So many targets of quorum sensing are clusters of genes that are typically associated with a given function. This is, as you remember, is called an operon. And one of the interesting things about being virulent bacteria is that most of your virulence genes, if not all of them, tend to be clustered together in these operons. So they're typically transcribed together. And what's sort of interesting, operons can exist and generally do exist in non-pathogenic bacteria, but many of these non-pathogenic bacteria can actually become infectious under the correct conditions. And this is what we call an opportunistic pathogen. And so if you ever heard this sort of term in a clinical setting, it's sort of a pathogen that just sort of, it's not something we typically think of as a pathogen, but just happened to say pop up because the patient was, say, immune compromised. And so one of the best examples of this is actually Pseudomonas aeruginosa. It's typically a benign soil bacteria, but can cause deadly lung infections. And one of the ways it can do this is actually by using quorum sensing. And then finally, so we can ask the question of why wait to turn on virulence genes. And one of the reasons for this is simply because our immune system typically keys in on pathogenic compounds. And so if you wait till you're high enough density to sort of survive the onslaught of the human immune system, you could potentially become a better pathogen overall. And so just to give you examples of this, so Pseudomonas aeruginosa, as we've talked about, is a normally safe bacteria, and it's something that can be very deadly. It's one of the top 5 most antibiotic resistant bacteria in the world. However, those that have sort of compromised lungs or weakened immune systems or have a condition called like cystic fibrosis, they can actually get Pseudomonas aeruginosa infections in the lungs, and these lung infections actually form as biofilms. Sort of what's interesting, if there's only a few pseudomonads in the lungs and they release toxins, they would essentially get easily destroyed by our immune system. However, what Pseudomonas does is it waits till it gets in high enough cell density, and it senses its cell densities again through these AHL compounds. And once it's in high enough cell density, it turns on virulence factors, and then it infects a large amount of the host. Next up is Agrobacterium tumefaciens, and this is actually a sort of a agro um, as well as environmental pathogen. So if you've ever sort of been walking around maybe in the woods or even in the city of Boston, you'll see trees that have like these little tumor-like structures growing on them that can be kind of big. This is caused by Agrobacterium tumefaciens. They're typically what we call galls. And so when Agrobacterium infects a plant, its quorum sensing pathways essentially encourage the transfer of DNA between cells, so within Agrobacterium cells. And essentially what this does is it promotes the transfer of virulence factor that make it much more pathogenic. And essentially it uses, the idea being is it only activates this quorum sensing when there's enough bacteria around. So it's only going to transmit this information, this DNA around if there's enough bacteria, and it does this by quorums. It senses this by quorum sensing. Next up is Erwinia caratova. This turns your vegetables brown in your refrigerator. So I just threw out a bunch of celery earlier this morning because of this bacteria. But like Pseudomonas, it waits until there's enough members to cause an infection. And once there's a sufficient number, they release proteases that cause a wound. And then it also will release antibiotics to kill other bacteria, allowing it to keep the wound all for itself and thus all the nutrients associated with said wound. All that brown stuff. I should also mention this is a different brown than, say, the browning of like bananas, which is just due to oxidation inside the banana. And then finally, we'll talk about Vibrio cholera, and we'll talk about Vibrio, and a very close relative of cholera, Vibrio fischeri, in a moment. But Vibrio cholera is sort of weird. It keeps all its virulence factors on at a low cell density, and this is what makes you sick. Its quorum sensing pathways do lead to the microbe shutting down its virulence machinery and focus on detachment from the intestinal lining. The idea being it does this so that it can make you sick, get in high enough cell density so that it can then detach and then go out into the world and infect other hosts. And it's sort of interesting, Vibrio was directly in contrast to most pathogens because they use their quorum sensing to stay and persist, while Vibrio uses it to physically escape and infect more hosts. So, fun fact. And so we're actually going to see a really cool example of this, and we'll just start off with this very nice picture of this glowing bioluminescent flask. And this is caused by a bacteria called Vibrio fischeri. It's a free-living marine bacterium, but when it gets in very high cell densities, the whole culture spontaneously glows in a process called bioluminescence, as you saw in that previous slide. This coordinated phenomena was recognized as density dependent and became the first research model for quorum sensing. And it's actually kind of interesting. So you remember that lux I and lux R. Well, lux translates to light, which is why they're named Lux I and Lux R. And so why is this cool? Well, this is the interesting thing about this is that we would never see high enough densities of Vibrio fischeri in the environment. So the question is, where do we see this? Well, the cool thing is we actually see it inside the Hawaiian bobtail squid, this beautiful little gastropod here. And so the Hawaiian bobtail squid is a small nocturnal squid. It lives in shallow waters. It hunts at night. And so the moonlight and stars cast a shadow. And one of the interesting things that predators will do is they look for shadows when they're trying to hunt. And so how does the bobtail squid avoid casting a shadow? Well, it does this by making its own light. And so what this squid does is it maintains colonies of Vibrio fischeri in special sacs that emit light when they reach a high enough cell density. And so it's kind of interesting. The density of Vibrio fischeri inside a bobtail squid can reach up to 10 to the 11 per milliliter, which is more than a typical lab culture. And so when we know what we've been doing in lab thus far, we're talking about 10 to the 9 bacteria per milliliter. We're seeing 2 orders of magnitude or 100 times more bacteria per milliliter inside of the bobtail squid with Vibrio fischeri. We actually don't know how they do this yet or how they maintain it, but they do at the moment. And so this is actually a pretty heavily studied system simply because it's a really simple way to understand how quorum sensing works and an actual meaningful function to study. So with that, we'll summarize the first part of this lecture. So bacteria can communicate with each other via density-dependent signals called autoinducers. Quorum sensing allows microbes to perform coordinated behaviors, and there are different autoinducers that allow for within species and between species communication of our microbes. And then quorum sensing pathways are important for formation of biofilms, which we're going to talk about in a moment here, virulence, symbioses, and as well as affecting the ecology of microorganisms as a whole. So with that, we're going to hop on to part 2 of this lecture, which is going to be biofilms. And this is a really important clinical part of this, clinical aspect of this lecture. And so we're going to answer the questions: what are biofilms and what are they made out of? What are the steps by which biofilms form and how are the stages coordinated? And what are some of the advantages to living in a biofilm? And so what's a biofilm? Well, a biofilm is, at its basic textbook definition, is a structured community of cells enclosed in a self-producing polymeric matrix and adherent to an inert or living surface. So there are 3 components here: microbes, slime, and a surface. And this is, when we're thinking about biofilms, this is what we sort of think about when we're looking at a culture or we're looking at sort of a picture of microbes. This is what it looks like in principle. So microbes growing together on a surface. So you can see the disgusting dental plaque here. This is a biofilm of microbes. Or if you've ever been to a stream and you sort of stepped on a rock and it's all slippery, that's a biofilm on a surface. And so microbes, as far as we know, most of them exist in biofilms. They're found in aquatic systems. So as you'll know next class, when we talk about marine systems, there are tons and tons of particle-associated bacteria, and they're all on those particles. And the vast majority of bacteria on those particles are actually in a biofilm. Sort of the other interesting statistic you can find about biofilms is that we potentially think that 99% of all bacteria do exist in biofilms, which is an absolutely astounding number of microbes out there. Many species of microbes do make biofilms. They exist across the domains of life. So we know they exist in archaea. We know they are found for bacteria. We know that when people have yeast infections, they're fungal biofilms. And we also know that pond scum is also a eukaryotic biofilm. And the clinically important thing is that these microbes are actually, in biofilms, are actually associated with 60 to 80% of all infections. And they're particularly important when we're thinking about surfaces like catheters and stents embedded in patients. Also, respirators. Fun fact if you've been following any of the stuff about COVID-19. But biofilms are a really, really old thing. There's evidence that they date back to 3.4 billion years ago in the South African Kronberg Formation. We also can find them in fossilized dinosaurs. And so this is a fossilized dinosaur bone, and this is actually bacterial biofilm. And one of the sort of interesting things about this is that they actually used to confuse bacterial biofilms as soft tissue for these dinosaurs, but actually further analysis showed that this is actually a biofilm and actually not soft tissue. And then finally, the other place we know that they're really old is actually in stromatolites. So if you remember our talk about our our origins of life, stromatolites are extremely old structures, and we find biofilms inside these stromatolites, both historically as well as in modern stromatolites. And so there are four main characteristics of biofilms as a whole. So there's autopoiesis, the possession of the ability to self-organize; homeostasis, resisting environmental perturbations; synergy, they have to be effective in isolation, they have to be effective in association rather than in isolation; and then communality, they need to respond to environmental changes as a unit rather than as a single individual. So let's start off with autopoiesis, possession of the ability to self-organize. And so this leads us directly to talking about biofilm formation. And there are multiple stages associated with biofilm formation, including reversible and irreversible attachment, maturation, and then finally detachment. And so, and this is sort of just a schematic. We have attachment and colonization, growth and proliferation, and then maturation and detachment. And so initial attachment is reversible, or it's also called a primary adhesion. And so for cells to form a biofilm, they need to first physically stick to a surface. And it needs to be contact between the microbe and the surface. And so this can happen randomly. So you can think of like, the water of a stream propelling a bacteria to touch a rock, or your bloodstream propelling a bacteria to touch, say, the artery of your heart, or by a bacteria settling out of a liquid. It can also be promoted by chemotaxis, or motility of the bacteria towards the surface. But ultimately, once the bacteria gets close, so less than 1 nanometer, just to put that in perspective, there is 10 to the 9 or 1 billion nanometers per meter. So this is a very, very small scale. So once the bacteria gets very close, the attachment results from the net sum of attractive as well as repulsive forces. And these are typically things that we think of when we think of attractive and repulsiveness, such as electrostatic forces, so positive, negative charge things, as well as things like hydrophobic and hydrophilic interactions. And there's also this also important factor called van der Waals forces, which is pretty weak, so we weren't really going to talk about it very much. That being said, our bacteria are typically negatively charged, so teichoic acids and gram-positive make them negatively charged, and lipopolysaccharide make gram-negative negatively charged. But the initial attachment, just as a note, all surfaces are able to be attached to. So even those surfaces that are specifically engineered to not develop biofilms can actually have biofilms formed on them. So pretty much anything a bacteria wants to attach to, it will. But bacteria are typically selective about where they want to attach. So for instance, members of the genus Pseudomonas, so aeruginosa and fluorescens, will form biofilms under almost any condition. Some strains of E. coli and Vibrio cholera will not form biofilms in minimal media unless they're supplemented with amino acids. However, other E. coli strains make biofilm only in low nutrient media. But other environmental cues that can influence bioformation include temperature, osmolarity, pH, iron, and oxygen, and the list sort of goes on and on and on. So, and it does vary between different species of bacteria. But ultimately, these bacteria need to form a conditioning layer or film. And so the properties of the surface that they're going to attach to do matter. So surfaces that are rough and hydrophobic tend to get colonized faster. However, smooth and hydrophilic surfaces will also eventually get colonized. Proteins and polysaccharides will stick to any surface that has liquids in the environment. This is what we term the conditioning film. It develops particularly rapidly. So the idea being you could put a glass slide in a fish tank and it will develop a conditioning film within minutes. So it's something that happens very, very fast. And these conditioning films can completely mask the physical chemical properties of the underlying surface, and it makes the surfaces easier to develop a biofilm on. And so your teeth, for instance, are really get really easily colonized with this conditioning thing that allows the biofilm to form on your teeth particularly fast. So after they sort of do that conditioning or initial colonization, they form our secondary adhesion, or the irreversible attachment. So once the microbe has attached, they can further stick to this using proteins and molecules called adhesins. And the idea being that these loosely bound microbes excrete these very sticky exopolysaccharides, so these just sugars that complex with the surface. In addition, flagella, fimbriae, and pili, if you remember our bacterial structures lecture, are also important for attachment to surfaces. And there has been some work showing that mutants that don't have these actually show decreased ability to attach to surfaces. But ultimately, once the microbe is stuck, it's there permanently, unless it's physically or chemically removed. So you can think of like when you brush your teeth, you're um, physically removing biofilms from your teeth. And then finally, once it's bound, it can act as a landing spot for other microbes. And so it can actually promote the attachment of other species, which will ultimately contribute to self-organization. So we can just look at just a basic schematic here. We have that primary adhesion, and then we start to produce these exopolysaccharides. Which encourage cellular aggregation. And then we have finally sort of the production of slime, sort of making the sliminess of biofilms. But in terms of irreversible attachment, an organism can have multiple means to attach. So for instance, Vibrio cholera has three methods, and it depends on where it's colonizing. So Vibrio cholera has what we call a type 4 pilus. It's important for colorizing the gut of animals, and thus it is an important virulence factor. It has a second type 4 pilus. That's not required for gut colonization pathogenesis, but it does aid in it, but is required for the biofilm formation on abiotic surface, such as plastic or glass. And then finally, Vibrio cholera is also known to colonize the chitin surface of shellfish. And many shellfish are actually a natural reservoir for Vibrio cholera and chitin because they use the sort of the chitin surfaces of shellfish as a nutrient source. And so we actually don't know how Vibrio cholera colonizes the surfaces of shellfish because mutants in both of these MSH and TCP proteins that promote these two types of colonization that we know actually have no effect on the colonization on chitin surfaces. So it's sort of this third unknown mechanism. But once a colony adheres, the cells have to proliferate. So it has to grow, they have to divide, they have to get larger, and they have built out horizontally as well as vertically. And so we can think of just sort of bacteria landing, and they have to proliferate, but they don't just proliferate outwards, covering the surface completely, but they also build complex 3D structures. And these act as essentially as you'll sort of notice, they can act as channels to allow for the flow of nutrients as well as waste products. And it commonly forms mushroom shapes without any currents as well. But these, they might look random, but they are highly, highly organized. And just to sort of show you what we're seeing here, so this is just an electron micrograph. Each color in this is actually physically just two different bacteria. And what you notice is that they're organized non-randomly. Well, maybe that's not the easiest thing to see, but just take my word for it. They're organized non-randomly, with the red ones typically more on the outside and the green ones typically more on the inside. And so maturation typically does follow a pretty predictable pattern and a particularly organized level of structure. And you can physically look at different sort of depth profiles in this biofilm, and you'll notice as we go deeper and deeper, there's more and more oxygen. And it's simply because of the structure of this biofilm, the way it's physically formed, both vertically as well as horizontally. In addition to the sort of 3D structure, we also, another important part of maturation is the recruitment of other organisms. As I mentioned, most biofilms are not just one organisms, they're typically multi-species communities. And so we can see just a very basic example here. We have a tooth microbial community, we have the tooth surface, as well as the salivary pellicle. And what you notice is we have 20 different types of bacteria all organized in a 3D structure. So we have, for instance, we have streptococci down here on the bottom, and then we have very different types of microbes up here. And they have a pretty, a very sort of varied amount. So pretty diverse group. But ultimately, the maturation of biofilm and the recruitment of new microbes is typically coordinated by quorum sensing molecules. And if you're trying to recruit, say, another streptococcus to your biofilm, you can use something that's specific to streptococcus. However, if you're thinking about trying to recruit something from a potentially different phylum, like say the spirochete right here, you need to use one of those much more broad reaching AHL autoinducers to recruit other microbes to them. Microbes are typically recruited in specific orders, and they're recruited by specific microbes. So for instance, Streptococcus might not be able to recruit every microbe present in this diagram, but maybe it can recruit these three because it's generally found closer, and maybe this microbe can recruit, say, these ones because they're related, and this microbe can only recruit this microbe, and so on and so forth. So the maturation does follow a pretty predictable set of succession as these biofilms mature and become older. And then after these biofilms have matured, we follow this up by detachment. And so once the biofilm has begun to exhaust all the resources in a particular area, it will enter a death phase, and then ultimately it starts to initiate detachment. And this detachment process is characterized by the production of proteases and lipases, so enzymes that break down proteins and lipids respectively. And what this does is it breaks down the exopolysaccharides that slimy layer that is holding these biofilms together, upregulates the production of motor proteins evolved in motility. And again, this is all something that is signaled by quorum sensing pathways. So just as a basic sort of overview of what biofilm formation looks like, we have the attachment, we have these microcolonies form, the production of that exopolysaccharide slime layer, we have the biofilm maturation, forming those 3D as well as those 3D structures vertically and horizontally. And then finally, we have that dissolution and dispersal. So once the microbe has exhausted all its nutrients, it then enters that, again, that death phase, releasing microbes to swim out, find a new surface, colonize, and repeat the process over and over again. But biofilms are heavily arranged, and so the location of an organism within the biofilm is highly regulated. And one of the ways to arrange this is actually by the metabolic or environmental needs of an organism. And the best way that we know that this happens is actually through the study of dental plaque. And so that gross stuff that's on your teeth if you haven't brushed in a while. And so, for instance, we can just look at the basic tooth structure, and what you'll notice is that, and this is the outer layer of the biofilm growing out, and what you'll notice is that depending on which layer you're in, you have different microbes, and you also have different environmental communities. So on the outside layer, there's oxygen. On the inside layer, it's completely oxygen devoid, and thus we have the stratification of microbes depending on whether they like oxygen or hate oxygen. But what you also notice is they're organized to exchange compounds with one another. So they intake saliva and sugars, and they have the cross release and sharing of byproducts, whether it's CO2, lactate, or acetate. And depending on where you are, you have the access to different compounds, whether it's natural from the environment or produced by one of your biofilm friends. So we should briefly talk about EPS, or exopolymeric substances. These are typically referred to as slime. This is actually a scientific term, not just me calling it gross. Most exopolysaccharides, mostly exopolysaccharides and proteins, but there is also DNA, lipids, and humics inside of these EPS substances. Much of the advantages of a biofilm are actually a product of these exopolymeric substances that are excreted by these members. Interestingly enough, what these do is form a sticky impermeable barrier. And one of the sort of interesting things we can actually look at is how the production of these different exopolysaccharides influences the movement of substances inside. And so we'll talk about this much more in depth later in the lecture, but what you'll notice is that different biofilms that are being produced have differential susceptibility to alcohol. And we'll talk about this much more in depth in a moment. Interestingly, up to 90% of all the mass of biofilm can actually be EPS, and suggesting only 10% is actually physical cellular mass. And as we've been talking about, EPS production is induced by quorum sensing signals. EPS can actually be very useful for other stuff outside of biofilm production. So for instance, alginate, which is produced by Pseudomonas aeruginosa, is actually used in live casting in dental impressions. Hyaluronic acid, which is produced by Streptococcus, is actually used in wound healing and as a cosmetic additive. It's called dermal filler. You probably know more about that than I do. Dextran, which is actually used in medicine. Antithrombotic, one of the WHO's 70 essential medicines, and it's actually used in baking. So panettone, which is this gross cake that I don't know why people like, but they do. It's made by a microbe called Streptococcus mutans, and it's typically a plaque-inducing bacteria. It actually helps make your gums extra bleedy. If you've ever been to the dentist and your gums bleed, or if you're flossing and your gums bleed, you can attribute it to Streptococcus mutans and the production of dextran. xanthan or xanthan gum, which is named actually after the bacteria that produces xanthomonas. It is a food additive. You eat it all the time. You just don't really know it. It makes your food thicker, and it's made by bacteria commercially. So they physically don't synthesize it. They actually use xanthomonas cultures to make xanthan gum to use in your food. And then finally, pulilan. I believe that's how you sell it. It's spell it. It's produced. I'm sorry, that's how you say it. It's produced by the fungus Uriobastidium pollinus, and it makes edible films. And this is actually what we use to make those oh so intense Listerine breath strips. So there is lots of commercial uses for exopolymeric substances. So that's apopoiesis. Next, we're going to move on to homeostasis, the capacity to resist environmental perturbations. And so homeostasis, again, is resisting these perturbations. And what ends up happening is these biofilms insulate themselves from outside conditions, limiting diffusion and the trapping of nutrients. And these biofilms are actually remarkably waterproof. They have this capacity of called non-wetting, which resists aqueous toxins as antibiotics. And so we're going to talk about this concept of wettability because it is a really important thing to understand in terms of how biofilms work and why they're so tough to deal with clinically. So wettability is simply just whether or not a substance can physically absorb a liquid. So something like, your shirt can easily absorb water. Something like a piece of paper cannot very easily absorb water. Something like wood is not very good at absorbing water. And so we can physically just look at this. We have two solid surfaces here. And we can ask the question, which of these surfaces is more wettable, i.e. less waterproof? And I think the obvious answer is that when water beads up on a surface, so thinking about like on a table or like on a leaf, that means the surface is less waterproof or is more wettable. That's in contrast to here. It has less waterproof, and thus the water is more likely to be absorbed and spread about. And so we can actually physically measure wettability essentially by measuring these contact angles. And so all we physically do is just measure the angle of this vertical angle here that we can measure. And so in this case, we have a non-wetting substance. It has an angle greater than 90 degrees. We have a wetting substance that is less than 90 degrees. And so we can actually look at a real life example of this. And so this is the percentage of ethanol. So ethanol is a really commonly used sterilizer. It's really commonly used in laboratory settings. And so as we're going from 0 to 100% alcohol, we're moving up in how much ethanol we have. And then we have contact angle. Remember, high contact angles mean it's a non-wetting surface. And what we can look at is a couple different we can look at a wild type of a biofilm, and then we can look at PTFE, which is an industrial polymer used to coat many different things. So they're it's like almost a plastic-y substance. And you can see that the contact angle of PTFE starts really high, so it's not very very wetting. And as you increase the amount of alcohol, it becomes much more wetting as you increase the concentration. So the capacity for it to get wet is increasing. What you see for a biofilm, in this case, we have a streptococcus here. We see that it's essentially pretty much flat from 0 to 60% alcohol, so it's not a very wetting surface. But once we hit about 70% alcohol, it becomes a much more wetting surface. And so you can see that if you had a streptococcus biofilm, say on your table, and you try to treat it with 50% alcohol, it would simply beat up on the surface of streptococcus. Whereas if you treat it with, say, 80% alcohol, it would actually physically penetrate and wet this biofilm and thus killing the biofilm. So that's wetting, and that's if you're ever working in a laboratory setting, you'll always see people cleaning their materials and their benches with 70% alcohol, simply because this is sort of like the best alcohol to best penetrate these substances. And so there are a lot of advantages to being resistant to wetting, and the more resisting to wetting, the more impervious a biofilm is to chemical attacks. This includes antibiotics, disinfectants, toxins, heavy metals, et cetera, et cetera. And And so, for instance, we can look at a stack showing a Bacillus subtilis colony, and what we can see is that the liquid is failing to physically penetrate this colony because it just simply doesn't have the right contact angle, or it's not wettable with that substance. Or in contrast, this substance here, it easily wettens and allows the stain, so it allows the compound to physically move inside the cell, having more wettability here. So that's homeostasis. We'll next move over to synergy, the capacity to be more effective in isolation. In association rather than isolation. And so synergy, essentially, you're better with the group than as your own. And I hope many of you are like this on your microbe wiki project, where you're working really well together as opposed to working on your own. But essentially, this gives you some benefits with predator evasion and toxin formation, as we talked about with Chromobacterium already. It allows enhanced genetic exchange of genetic material. So we're thinking about horizontal gene transfer and the acquisition of new traits, including clinically relevant things like resistance to antibiotics, as well as pathogenicity, or more environmental things such as increased metabolic capacity. And it finally allows microbes to colonize places that normally wouldn't be able to. For instance, anaerobes being able to live inside your mouth, whereas your mouth typically is all sorts of oxygen rich. Anaerobes can colonize your mouth through the use of dental plaque. Antibiotic resistance is a really important thing to think about in contrast to biofilms, because as we mentioned, biofilms are really important in a clinical setting. So when we think about antibiotic resistance or sensitivity to it, it's typically measured in cells in a liquid culture. So remember when we talked about calculating that MIC, or the minimum inhibitory concentration, remember that's the lowest concentration of a substance that arrests the growth of a microbe. Or the minimal bacterial cytoconcentration, which is the lowest concentration needed to kill a microbe. Remember, these are always calculated on cultures of liquid, planktonic cultures. And so the MIC of a microbe growing in a biofilm can actually be 2 to 100,000 times, 2 to 1,000 times greater than the cells growing planktonically. So it's particularly hard for antibiotics to work on biofilms, and can make getting rid of biofilms in a clinical setting very, very difficult. But cells can become resistant because of mutations affecting the drug target. Essentially, we're thinking about efflux pumps. We can also think about the production of modifying enzymes. So this is how bacteria naturally become antibiotic resistant, and we covered this in our disease aspect of our lecture. So from 2 weeks ago, or one or 2 weeks ago, and we'll also cover it again in our global health portion of the course. But any cell can do this, but planktonic or in a biofilm. But cells that are in a biofilm are even better. And the answer to this question of why are they better is actually still up for debate. And so there's a couple of thoughts here, is that the structure of the biofilm may restrict the penetration of an antimicrobial agent. It's just as a note, once drugs many drugs are charged, and they can essentially bind to oppositely charged polymers that make up the biofilm matrix, thus not allowing them to penetrate the biofilm. The drug may also stick to and inhibit the organisms at the surface of the biofilm, but not get things deeper down. And then the matrix of the biofilm itself can bind and retain neutralizing enzymes. So we're thinking of something like a beta lactamase that might inhibit these drugs. So these microbes might inherently produce things that won't allow these antibiotics to get deeper down, and thus fully disrupt the biofilm infection. But there's even more here. Biofilm microbes also display novel phenotypes, which may particularly lead to the reduced sensitivity to these inhibitors. So for example, drug targeting can be modified or not expressed, or can be used via alternative metabolic strategy. Cells in biofilms often will grow slower in the nutrient-deprived conditions of a biofilm, and thus are less susceptible to many antibiotics relative to, say, faster growing ones that are not in biofilms. And finally, the environment in the depths of the biofilm may be actually unfavorable to the optimal action of some drugs. So for instance, pH can have a pretty dramatic effect on the efficacy of any given drug. And many times deeper down you go in biofilms, the more acidic it gets. So thus that even if the antibiotic could get down to the deeper layers of the biofilm, it simply wouldn't be as effective because of the pH. And so let's wrap this second part up. And so what we want to take away from this is that biofilms are organized communities of microbes and consist of microbes by themselves, a sticky exopolysaccharide, and the surface that they're on. Biofilms do progress through stages of initial and permanent attachment, maturation, and then finally dispersal to go on and form new biofilms. These steps are mediated typically by quorum sensing, but they also can be affected by specific receptors on a cell surface. Biofilms are characterized by having autopoiesis, homeostasis, synergy, and communality. And then finally, biofilms can confer many advantages to these microbes, including this capacity to resist toxins, wetting, as well as antibiotics. And so part 3 and the final part of this lecture is going to be the second part of biofilms. If you're a fan of Star Wars, that reference at the top will make sense to you. So we can ask the questions today. What are some of the advantages of living in a biofilm? How do successional patterns in biofilms happen, and what coordinates their progression? And what are some factors that affect biofilm activity and composition? So as we mentioned, there are numerous advantages here, in particular with wetting, in terms of preventing the proliferation of antibiotics or disinfectants or toxins. But microbes are also more resistant to wetting, and the more resistant a biofilm is to wetting, the more impervious a biofilm will be to chemical attack. And so, for instance, we have a colony here of bacteria, and it's been treated with bleach for 60 minutes. Colonies that are green are dying. Those that are red are still actively growing. And what you'll notice is that most of the cells in here are actually still living, even after being treated with 60 minutes of bleach. And we can also look at another sort of example of this. Again, green is live, red, yellow is dead. And we're looking at furanone. And furanone is a compound that does interrupt quorum sensing. And what you'll notice is that we have, again, dead and alive in green. And you'll see that no furanone, not having any meaningful effect, but if we treat them with 10 grams per mil of furanone, what you'll notice is that there's a huge impact on who lives and who dies. And so furanone, what it does is it actually physically disrupts quorum sensing by modifying signaling molecules. This is an example of an AI2 inhibitor. This is a compound that comes from algae. The algae is Decelea percha, or a red algae. It makes 30 different types of furanones, which is actually used to limit the growth of bacteria on the physical surface of this algae. It limits the ability of any microbe using the AI2 system, which, as we've talked about, can be which can be a lot of different microbes. And sort of one of the interesting things that some of the other furanomes will disrupt AHL-mediated quorum sensing as well. And so let's talk a little bit more about this concept of synergy. So this is the capacity to be more effective in association than in isolation. So as we've talked about, there's a number of different properties here, including predator immune evasion, toxin enhanced exchange of genetic material, as well as the capacity to colonize area that you wouldn't be able to. In addition, it provides numerous benefits of antibiotic resistance, increasing the capacity of a bacteria to withstand things. It also modifies the general capacity of a bacteria to be resistant to antibiotics. And again, as we've mentioned, the antibiotic resistance question is still up for debate. Why do biofilms become more resistant? That's still an open-ended question. And there's a lot of sort of things that come into play here when we're thinking about how biofilms are antibiotic resistant. But ultimately, biofilms, when they're in an environment, whether it's a clinical or just sort of a regular environmental setting, our biofilms are multicellular organisms, essentially. They respond, and they need to respond to changes in the environment as a group, not just as a single individual. So this can manifest as changes in community composition of the biofilm itself. So say they get exposed to, say, bleach, they can change the composition of the community, or they can simply change the activity or gene expression of the community as a whole. But again, this is something that is still coordinated. So for instance, exopolysaccharide production is best around a pH of 7. So highly acidic or basic environments will limit biofilm proliferation. In addition, temperature does alter adhesion of microbes to a surface. So primary adhesion does depend on weak molecular forces. So at elevated temperatures, things are a little bit more difficult to do, and thus primary adhesion is harder at higher temperatures. In addition, that evidence does suggest that at low temperatures, adhesins are less sticky as a whole. But ultimately, as we've been talking about this course, the environment matters, not just for bacteria or viruses, but it matters for biofilm formation. So normally when we think about a potential pathogens here in gray, they need to be present or transmitted in low numbers. But a major ecological pressure might be necessary for a pathogen to essentially outcompete resident flora, represented here in white, and achieve the numbers required to cause disease. So we can think of healthy individuals we can think of two healthy individuals. One individual has the pathogen, but it's at very low numbers. It transmits to the other healthy pathogen. And then we have some major ecological pressure. It could be low pH, a sugarish diet, or low salivary flow that allows this pathogen to proliferate in this new environment. And so just to sort of highlight what's going on here, we just have this table. It's looking how glucose and pH affect the stability of a microbial community. So this is a sort of a basic example. We have 9 cultured oral bacteria that are representative of those found in healthy and diseased gum, and they're grown at a pH of 7. And what they did was mess the pH with the they messed the pH of this media to see how it would affect the growth of organisms. So for instance, we have Streptococcus mutans and Lactobacillus rhamnosus are disease causing organisms, whereas Streptococcus gordini and Fusobacterium nucleatum, are healthy. And what you'll notice is that at certain different pHs, we have different levels of biofilm for different percentages of viable cultures. So we have no pHs or very acidic pHs. We have very high amounts of these pathogenic ones, but at sort of lower levels of pH, we have essentially no pathogenic microbes, and we have very high amounts of these sort of healthy ones. And so that's synergy. And we'll sort of wrap up our talk about biofilms by thinking about communality as well as some other factors about biofilms. But remember, communality is coordinated behavior. And so remember, the presence of a biofilm does alter patterns of gene regulation as well as gene expression. And it can be affected by surface recognition as well as quorum sensing pathways. One of the interesting things that can happen in biofilms is we can see novel phenotypes come about by changes in gene expression. And these changes typically are rapid and substantial. So for instance, genes that are responsible for alginate synthesis in our friend Pseudomonas aeruginosa are upregulated within 15 minutes of contact with the surface. Or just another example, or 73 or in 50% of the detective proteome, so what proteins a microbe is producing at a given time, were differentially regulated in biofilms of Pseudomonas when compared to planktonic cells of Pseudomonas. But ultimately, these coordinated behaviors often affect pathogenicity. And so we could think about potentially some of these genes or proteins might be actually virulence factors of these organisms. And so we'll talk a little bit about Pseudomonas aeruginosa for a little bit, again, because this is one of our favorite microbes in this course. Actually, it's just my favorite microbe, but it's a really good microbe to sort of illustrate many different concepts across this course. But Pseudomonas aeruginosa in immune-compromised organisms will actually infect the alveoli, which are sort of these little pockets of your lung. And the idea being is that Pseudomonases that are caught in these little alveoli typically only occurs when your immune system is compromised. And so the idea being is that the biofilms do not trigger sort of your immune responses. And what ends up happening is it causes, as these biofilms sort of age and mature, it causes the biofilm to calcify, harden, and inactivate your alveoli. And once that happens, the idea being is that will essentially render that part of your lung essentially inactive or non-functioning. Another example of this is actually toxic shock syndrome. It's the result of the release of toxins by Streptococcus or Staphylococcus biofilm. And so when biofilms are under immune attack, when they can release as a defense mechanism the production of toxins. And one of the best known cases of this is toxic shock syndrome, which many of you have probably heard of before. But this is the release of toxins by strep and staphylococcus biofilms. And so infections can come from many sources, but often surgeries, implants, and other medical procedures commonly introduce infections inside the human body. This is something that is also pretty common among menstruating women. So bacteria can form biofilms on say tampons and the things associated with tampons in menstruating women. And the microbes that can colonize the vaginal tissue in tampon fibers can become potentially pathogenic. And they can, when they do develop into biofilms, they cause inflammation and diseases such as toxic shock syndrome. And just as a note, for those of you that like the Muppets, this is actually how Jim Henson died, from toxic shock syndrome. Tampons are a really common way to sort of think about this, but it can happen on anything that is inserted inside your body. And so we're going to dive a little bit deeper into biofilms, and we're going to do this by talking about one of the cooler things in microbiology, I think, but one of the gross things in my opinion. And not very many things gross me out, as you probably all know, but dental plaque actually does gross me out because dentists, teeth bacteria and teeth are just gross. So we're going to talk about dental biofilms, and we're going to think about how they work. And dental biofilms are really cool because they can have upwards of 500 species of microbes living together. They are typically sort of they're typically not that harmful, but they can cause some pretty severe diseases if left untreated for an extended period of time. But we've been using dental plaque as a model for biofilm formation forever. So if you remember back to it was one of our early lectures when we talked about von Leeuwenhoek and his animalcules, when he looked at his tooth scrapings under his microscope, he was looking at biofilms as these biofilms on his teeth as a model way. So we've been using teeth as biofilm models for a very long time. More importantly, and I hope you'll take away from this, is how important brushing your teeth is to your overall dental health. Or if you want to be like some people who don't like to brush their teeth, you can let them grow for fun and let them develop and let them do all sorts of cool stuff. So to be an oral microbe, you need to be sticky. You need to be able to stick to something in your mouth. And those microbes that can't physically stick get swallowed and they go to your stomach. And almost no bacteria can survive going through your stomach. So if you get sent to the stomach, you're pretty much toast. All normal microbes can either adhere to solid surfaces, essentially thinking about like your tooth or even your gums as well. They can also adhere to other cells. And so they can adhere to cells stuck to a surface. This is what we call coadhesion. They can adhere to cells in suspension, which is called co-aggregation. And these cell-to-cell interactions can be species level specific. So for instance, Prevotella lotioni and Streptococcus sanguinis typically will only stick to others like themselves or closely related things. And sort of one of the interesting things about oral bacteria is we know about 1,200 or so of them at this moment. They all have been shown to have at least one coadhesion or co-aggregation partner. And we can just sort of look at a basic schematic of what this looks like. And so we have early colonizers down here, late colonizers up here. And this is, again, what we've looked at this thing before, the tooth surfaces here. And what you'll notice is there's production of all sorts of different molecules by these microbes to help them stick to the tooth surface. But what you notice is that they have all sorts of lines linking them together because they're aggregating with one another or co-adhering with one another. And so tooth enamel itself is rapidly coated in a suite of sugars and organic molecules and proteins. This is what we call the dental pellicle, which acts as that conditioning film in the early stages of the biofilm. And after you brush your teeth, even if you use Listerine or anything, this will develop within minutes of you brushing your teeth. So even though you get rid of your microbes by brushing them, they come right back real fast. This layer does contain many molecules that initial colonizers recognize and complex with or adhere to. These include slatherin, proline-rich proteins, and alpha amylase, and a whole suite of other things. Key initial colonizers inside of your mouth include streptococci, and they particularly like to bind with amylases. We also have actinomycetes who like to bind to slatherin and produce proline-rich proteins. And then finally, fusobacterium, which recognize slatherin. I'm sorry, statherin, not slatherin. Pronunciation has never been my strong suit. And we can see this. So we have the production of, say, staphyrin, which binds to number 14, which is our fusobacterium. We have alpha amylase, which binds to one of our streptococcus, and so on and so forth. So we have this sort of co-aggregation and co-adhesion pairs. Then after that, sort of that initial colonization, other microbes need to adhere to the bacteria that are already stuck and build the biofilm out. So for instance, Fusobacterium nucleatum aggregates with the initial, early, and late colonizer, essentially acts as a bridge for later colonization of the biofilm. Just as a note, this is mediated by a surface proteins onside the surface proteins on the membranes of these microbes. So after we sort of have that colonization, eventually the biofilm needs to mature. So it needs to build and add new taxa as they continue to adhere and aggregate with their suitable partners. This essentially leads to stable and healthy biofilms on the tooth surface. That being said, we typically think of bacteria in a clinical setting to be bad, but remember, your microbiome is important for preventing the colonization of other microbes. And as long as you sort of keep your microbiome in sort of a check with your own body, you can prevent sort of bad things from happening. But your teeth do have a healthy biofilm. I know you might not think that, but you do have a healthy biofilm. So streptococci, which are the most abundant organisms in your biofilm, they do ferment low molecular weight carbohydrates. So you're thinking about like glucose, sucrose, things like that. They convert them to organic acids, which actually lowers the pH of your mouth. And then for those of you that know anything about dental, sort of the way your teeth work, this lower pH by streptococcus actually dissolves your enamel, which is that outer coating, which is actually kind of bad. Sort of interesting fact, this is what happens when you eat. So when you eat any food, the bacteria in your mouth go crazy because that's their only source of food. And so they do all this fermenting, it lowers the pH. But after you're done eating, the pH starts to rise again. So it's actually, if you talk to any dentist, they will recommend you chew a gum say Trident, which has xylulose in it, which actually helps bring the pH up really, really fast. Sort of later stages of maturation of your biofilm. So if you left your dental plaque for days and days and days without brushing, very old dental plaque does contain very large populations of acidic bacteria or acid-generating and acid-tolerant bacteria. This includes bacteria called Streptococcus mutans and Lactobacillus, and they produce much more acid than your sort of normal healthy bacteria. And they thrive in the world of low pH. So what this does is it creates this nasty positive feedback loop that increases their numbers and gives you cavities. So if you let a biofilm sort of mature for an extended period of time, say like in between your teeth, because those are sort of hard to brush and take care of, this is why you sort of start to get cavities, because these bacteria are producing these acids, they're breaking down your teeth, they're colonizing the breaking down parts of your teeth, causing holes, causing the further break down your teeth. And you sort of let these go for an extended period of time, they'll eventually get down into your sort of, your dentin, so the inner parts of your teeth, and eventually your root, and eventually you need a root canal or you get the teeth pulled. And so this is sort of the transition period. As it matures, it starts to lead to much more deleterious things. But that sort of initial group, that sort of that young biofilm on your teeth is actually good. But that being said, your biofilm, like all biofilms, are coordinated. So dental plaque does provide an efficient place for cell-to-cell communication inside your mouth. And this is really, this is something that's important because there's essentially no time to build up any sufficient concentrations inside your mouth. So they need a place to sort of get in higher numbers, and that place is on your teeth themselves. There's evidence of abundant crosstalk between microbes using peptides and AHLs inside of your mouth. And there's also been shown interkingdom crosstalk between Staphylococcus and Candida, which is a fungus. And one of the interesting things is it actually induces Candida to produce more hyphae, which allows the biofilm to get larger. So our autoinducer 2, or AL2, seems to be the dominant communication system within our dental biofilm themselves. Sort of interesting thing is if you have mutants you have bacteria that are mutants in these AI2s, they actually fail to generate biofilm. So these this quorum sensing does seem to be important for the development of biofilms inside your mouth. And so we can look at just sort of a very basic structure, very basic overview here of what's happening inside your mouth. So we have that initial association and adhesion. Then we have the proliferation. So as you notice, we're getting bigger and larger. But you can also notice is that we're starting from sticking directly to the surface of the microbe to then start building upon other microbes. And as you'll notice is that we go from proliferation to colonies, we're getting taller and taller and taller. We form the biofilm, and then we finally have that maturation. Remember, when a biofilm is mature in dental plaque, it does two things. One, it's sending microbes out to form new dental plaques, and it's also producing much more acid to start to dissolve your teeth as a whole. But these biofilms are great as model organisms to understand how biofilms, because all the processes that occur in biofilms occur inside dental plaque. And there's all sorts of cool, so this is just wrap up this lecture by showing you some really cool sort of 3D pictures of how these biofilms organize. So this is a really old picture of your tooth and just sort of a nice, easy micrograph, like something you would take in lab of what a biofilm looks like. But as I've shown you before, these biofilms and these plaques get pretty crazy in terms of the microbes and how they organize. So you'll notice that, you know, in all these cases, typically the tooth is on the bottom or on the side over here. What you notice that there's and each one of these colors here is a different microbe. So like, for instance, yellow is a fusobacterium, green is streptococcus. And you'll notice that there's a high degree of sort of order within these biofilms, where you have different organisms lining up the exact same spot with each other. And it actually goes along really nicely with sort of this sort of structure we're thinking about here. You can see it in real time. And again, you can see again here. Again, pretty pictures of these microbes organizing in 3D. And again, all mediated by quorum sensing, as well as adhering to one another and forming clumps. And then finally, sort of a big picture thing. This is a much bigger, so you're talking, you know, this is almost a millimeter long in terms of this picture. And you can see it happens both as we've sort of been looking at the very small scale, but also at the very large scale. And I say large scale for microbes. Right? And so with that, we're going to wrap up this final section of the lecture. So biofilms confer many advantages to these microbes, including resistance to toxins, wetting, and antibiotics. Biofilms can be affected by and respond to their environment, and this can manifest itself as changes in gene expression or shifts in community composition. Dental plaque itself is a great example of having a biofilm, which highlights many key concepts of biofilms as a whole. If any of this sort of talk about this dental plaque interests you, please let me know. There's tons and tons of literature out there about this dental plaque, and it has all sorts of cool pictures like this, which I think makes microbiology fun. But that will be the end of our lecture for the day. Please let me know if you have any questions, and I'd be more than happy to answer them. And I hope you all do well. Take care.